Hello everybody, welcome to today's lecture on biochemical reactions with the focus being on cell growth. In the last lecture we talked about enzymatic reactions and how to perform material balances for systems where enzyme reactions are taking place. Today we will talk about cell growth and the fundamentals associated with it and perform some material balance calculations for systems where we have cell growth. First, let us learn some of the terminologies associated with cell growth. Growth rate is defined as the rate of change of cell mass concentration, which is represented as x, and the units is usually grams per liter. This is given as growth rate is dx dt. So, this is the mathematical representation. So, we need to understand that this is an extensive property, which means it depends on the size of the system. So, if you start with 1 gram per liter cells and the doubling time is 10 minutes, after 20 minutes you would have 4 grams per liter of cells, whereas if you start with 10 grams per liter cells to initially, after 20 minutes you would have 40 grams per liter and your growth rates are technically different. This is because of the initial inoculum size that has been used. To eliminate this difference, we use something called specific growth rate. This is an intensive property, and mu net, which is the specific growth rate, is given as 1 by x dx dt. So, this is an intensive property and we can compare cultures without worrying about the inoculum size which was used for growing the cell. We can compare cultures without worrying about the inoculum size that was used for the experiments. These terms can also be represented in terms of cell number concentrations instead of cell mass concentrations. The specific growth rate which we look at here is the net specific growth rate. We should remember that there is another term called gross specific growth rate, which is the net specific growth rate plus Kd, where Kd is the rate of cell mass loss due to cell death or endogenous metabolism. So, this in addition to mu net gives you the gross specific growth rate. There is also death rate and specific death rate, which can be defined similar to growth rate and specific growth rate. You have yield coefficients, where growth yield would be the ratio of biomass generated to the substrate consumed, and product yield would be the ratio of product formed to the substrate consumed. You also have a term called specific product formation rate. This is directly proportional to the gross specific growth rate for growth associated products. So, this is usually represented as QP, and this QP is equal to ypx times mu g for growth associated product formation. Ypx is the yield of product per unit biomass which is produced. Constant value of specific product formation rate will be observed for non growth associated product formation. So, that is why Qp would be equal to beta, a constant for non growth associated product formation. In case you have a mixed product formation, where you have some non growth associated products and some growth associated products being formed simultaneously, then the specific product formation rate Qp would be given as alpha mu g plus beta. When you culture the cells in a batch system, what you see is unicellular microorganisms go through different phases of growth. The first phase is the lag phase. This is the initial phase where the cells are getting adjusted to the environment, and there is actually no cell growth during this period. So, during this time the cells are getting an understanding of what nutrients are available, what are the conditions in which they are being cultured, and they prepare the machinery that is required for surviving in that environment and utilizing this nutrient to eventually grow and multiply. The next phase is the log phase or the exponential phase. Here the cell numbers increase exponentially and the cells are dividing rapidly. This is also a period of balanced growth, and during this period the cell growth follows first order kinetics. The next phase is the stationary phase. Here the net growth rate is 0, which is probably because death rate is equal to growth rate. Here the cells are still metabolically active. The non growth associated products are actually produced during this stationary phase. So, these non growth associated products are also called as the secondary metabolites. The stationary phase is followed by the death phase. During this time, due to depletion of nutrients and accumulation of toxic products, the cells start to die. Because of this, the cell concentration starts declining, and this period is called as the death phase. In addition to these 4 distinct phases, In some textbooks they also list acceleration and deceleration phases. Acceleration phase is the phase between the lag and the log phase, and deceleration phase is the phase between the log and the stationary phase. So, what you see here is the growth curve. So, you have the lag phase initially where the cells are getting used to the new environment and the cell numbers or concentration is not increasing. You then have an exponential phase where the cells are multiplying and the cell concentration is increasing rapidly. Then you have the stationary phase where the cells are actually dividing and also dying, thereby maintaining the same cell concentration. And finally, you have the decline phase or the death phase where the cells are dying either due to nutrient depletion or due to accumulation of toxic materials. So, the phase in between the lag and the exponential phase where you have the acceleration of cell growth, however, it still does not follow the exponential curve, is called as the acceleration phase, and the phase between the log and the stationary phase where it still does not follow the exponential curve, but it is starting to slow down, is called as the deceleration phase. One thing you should understand and remember when you draw a growth curve is you see that at induction the value for cell numbers is not 0, and this is important. Whenever you draw a growth curve you start with an inoculum. So, therefore, at time point 0 when inoculation occurs you are going to have a certain cell number which is then going to increase over the exponential phase. So, please do not draw a growth curve with 0 as your origin. Having understood the growth curve, let us move on to modeling cell growth. Unlike enzyme kinetics, cell growth is actually a result of various complicated reactions and networks. So, it is almost impossible to draw any kind of a model which will fully describe this system. At any given time the cell population is actually a mixture of young and old cells, and mathematical modeling would mean that there are going to be n number of parameters which are going to be very difficult to estimate. So, what people do instead is use a simple model which is the unstructured non-segregated model. We are going to look at one such model which is called the monods model. Before we do this, we should understand what assumptions have to be made for performing this type of an unstructured non-segregated modeling. The first thing is we will assume that cells can be represented as a single entity. It could either be cell mass, cell numbers, or concentration of a protein, DNA or RNA. So, the use of concentration of protein, DNA and RNA is acceptable for balanced cell growth period, which would be the exponential phase. Cells are assumed to be uniformly distributed in the culture. So, cell suspensions which are actually heterogeneous are considered to be homogeneous solutions, and the heterogeneity among the cells themselves is also ignored when we perform this type of modeling. As I mentioned, monods model is one such model which is very popular and it is consistently used for describing cell cultures. So, this is an unstructured non-segregated model which is used when you have a substrate limited growth kinetics. You assume that a single substrate is actually the growth limiting substrate and the modeling is performed based on that. The kinetics itself is similar to the Michaelis-Menten kinetics, and the equation for the gross specific growth rate mu g is given as mu m times S divided by Ks plus S, where mu g is the specific growth rate, mu m is the maximum specific growth rate, Ks is the saturation constant and S is the initial substrate concentration. So, when you actually culture E. coli with different substrate concentrations and measure the specific growth rate, this is how the graph would look. Specific growth rate versus substrate concentration graph looks very similar to what you would have observed with V versus S curve for enzyme kinetics. So, this is a saturation kinetics, and as you can see the specific growth rate approaches a maximum value mu max or mu m when the substrate concentration increases beyond a certain point. To perform material balances for cell culture systems, let us take an ideal chemostat. An ideal chemostat is a perfectly mixed continuous flow stirred tank reactor. So, the schematic is given here where you have F as the volumetric flow rate, S naught as the initial substrate concentration, X naught is the inlet biomass concentration. Usually you have a sterile feed and inoculum is just given as at time point 0 inside the reactor. So, your feed stream does not usually contain biomass. So, it would usually be a sterile feed giving x naught to be 0. P naught is the concentration of products in your inlet stream. So, which is also usually 0. F, S, X and P are the corresponding values in the outlet streams. You have a working volume which is the liquid volume inside the reactor, that is called as the reactor volume also given as VR, and you have S, X and P as the concentrations inside the reactor as they are the concentrations in the exit stream. Just like we discussed CSTR in an enzyme kinetics, here also the concentrations of components in the exit stream will be the same as the concentration of the components in the inlet stream. So, you have a well mixed system, which means whatever is fed is immediately mixed and the concentration is uniform throughout the reactor. In addition to the mixing conditions, you also see something called a gas sparge. So, this is usually present because bacterial cells need a lot of oxygen to grow, and this oxygen is supplied using a gas sparge, which is either oxygen, pure oxygen being supplied, or air which is supplied to provide oxygen to the cells to grow. So, let us now perform material balances for such an ideal chemostat. So, the first balance we are going to write is for biomass. Let us assume unsteady state and non-sterile feed for the time being, and then we will cancel out terms based on the assumptions which we make. So, let us start with the balance equation. Just like any material balance equation, your equation starts with input minus output plus generation minus consumption equals accumulation. So, the inlet term would be F times x naught, where F is the volumetric flow rate and x naught is the biomass concentration in the inlet stream, minus outlet being Fx, where F is the outlet flow rate and x is the biomass concentration in the outlet stream, plus generation term. We will get to what the generation term would be, minus consumption. Let us also get to consumption later. So, accumulation would be change in biomass concentration within the system with respect to time. So, that is dx dt, and because we want it in terms of change in biomass total mass with respect to time, we will multiply this with VR, or the working volume for the reactor. Generation for biomass will depend on the growth of the cells. So, that would be based on the specific growth rate, which is mu times x, which gives us the growth rate in terms of change in biomass concentration with respect to time due to cell growth. So, we want to get the change in total mass of the biomass or cells with respect to time. So, this again multiplied by VR, which is the volume of the reactor, will give us the generation term. Similarly, we can get consumption term as KdXVR, where Kd is the specific death rate or the specific rate at which endogenous consumption of biomass is happening. So, this would be KdXVR. So, substituting all of this, we get Fx naught minus Fx plus mu x Vr minus KdXVR equals Vr dx dt. So, you can also verify the dimensional homogeneity of this equation, just like we did for enzyme kinetics, and you would be able to confirm that this equation is dimensionally homogeneous. And when we consider a steady state operation, this term will go to 0, and if we do not have any endogenous reactions or cell death, your Kd will also go to 0. If you have a sterile feed, this x naught goes to 0, giving you an equation which is minus Fx plus mu x Vr equals 0. From here we get F by Vr equals mu. So, this is also equal to d, which is the dilution rate. We already know that F by Vr is the dilution rate. So, what we have here is, by altering the volumetric flow rate to the system, we can alter the specific growth rate of the cells. So, this makes chemostat a very useful tool for performing biochemical reactions. In addition to writing the biomass balance, we can also write a substrate balance. Let us try to write a substrate balance for the ideal chemostat. So, again we start with input minus output plus generation minus consumption equals accumulation. So, we will initially assume that it is not at steady state, and then we will cancel off terms based on making the appropriate assumptions. So, the input would be F S naught, output would be minus F S, and here we will not have a generation term because substrate is not going to get generated in the system, it is only being consumed. So, we have consumption. So, we will get to the consumption terms equals accumulation, which would be very similar to whatever we looked at with biomass. So, it is VR ds dt. So, now for consumption terms there can actually be two different consumptions, we will call them consumption 1 and consumption 2. Consumption 1 would be the consumption of substrate for the cells to grow. So, that would be the growth associated substrate consumption. In addition, you can also have product formation which could result in consumption of substrates. So, consumption 1, which is associated with the growth of the cells, would be given as mu x Vr divided by Yxs. What you see here is mu x Vr is the biomass which is generated and Yxs is the yield of the biomass with respect to per unit of substrate consumed. So, mu x Vr divided by Yxs will actually give you the amount of substrate which is consumed per unit biomass which is produced. So, the second factor, second consumption, which is for product formation, can again be written in similar fashion, which will be Qpx Vr divided by Yps, where Qp is the specific product formation rate and x is the biomass concentration. Qp times x will give you the product formation for the growth associated proteins and growth associated products, and you have Vr multiplied to get the total amount of such products being formed, and Yps is the yield of the product per unit substrate being consumed. So, thereby we can actually convert this into the amount of substrate which is actually consumed during the product formation. So, substituting all these terms back into the equation, we get Fs naught minus Fs minus Vr mu x divided by Yxs minus Vr Qpx divided by Yps equals Vr ds dt. Now, at steady state this term goes to 0 and your product formation, if it is 0, that would also go to 0, and your equation can be simplified to Fs naught minus Fs minus Vr mu x divided by Yxs equals 0. So, this is a balance equation for a substrate. While we perform these calculations, one thing you would have seen is the accumulation term for biomass was also given as dx by dt, and we had earlier defined growth rate also as dx by dt, but it is to be understood that these two terms are not equal except for batch reactions. In reality, change in biomass concentration due to many other factors other than growth can also happen. It could be because of change in flow rates, it could be because of accumulation of components and so on. So, that term is the accumulation term. The change in biomass concentration with respect to time due to cells dividing is called as the growth rate, which is also dx by dt. So, these two dx by dt's are not the same, they are numerically equal only for a batch reactor condition. Now that we have clarity on this, let us move on to performing some example problems to fully understand how to apply these material balance principles to biochemical systems where cell cultures are happening. Here is the example problem. Two related processes for growth of a bacteria have been proposed. Option 1 is to use a single well mixed vessel having a volume of 100 liters. Option 2 is to use two well mixed vessels each of volume 50 liters connected in series, so that the output of vessel 1 becomes the input to vessel 2. The relation that gives the growth of the bacteria in each vessel at steady state is the monod equation. The suspension contains water, nutrients which are called the substrates, and cells. Which option will give the highest concentration of cells in terms of grams of cells per liter? You have been given that the volumetric flow rate into the vessel 1 for option 1 and into vessel 1 for option 2 is 15 liters per hour and contains no cells, but it contains a substrate at the concentration of 10 grams per liter, and YXS is given as 0.2 grams of cells per gram of substrate consumed, and Ks is 2 grams per liter and mu max is 0.4 hour inverse. With all this information, let us perform the material balance calculations to identify which of the two systems will give us the higher biomass concentration. So, we will first start with the option 1. As we have been told that the system is operating at steady state and you have a sterile feed which does not contain any of the bacteria, we would have the biomass balance or the cell balance simplified to the equation mu equals F by Vr. So, this is based on the derivation which we did earlier. So, we can also write the substrate balance as we got earlier, it would be Fs naught minus Fs minus Vr mu x divided by Yxs equals 0. As we know that mu equals F by Vr, we can substitute the value for mu here and we end up with F times S naught minus S minus x divided by Yxs equals 0. So, this implies x equals Yxs times S naught minus S. From the values given in the problem, we can calculate mu as 15 liters per hour divided by 100 liters, which is the volume of the reactor for option 1, giving us a mu of 0.15 hour inverse. So, using monod equation, we know that mu equals mu max S divided by Ks plus S. So, we have been given mu max as 0.4 and Ks as 2. So, using these values we can substitute this as 0.4 S divided by 2 plus S equals 0.15, which is mu. From here we can calculate the outlet substrate concentration, which is S, giving a value of 1.2 grams per liter. Now that we know the outlet substrate concentration, we can substitute it back to the equation for X to calculate the value of biomass concentration in the outlet stream, which would be Yxs, which is 0.2 times inlet substrate concentration 10 grams per liter minus outlet substrate concentration 1.2 grams per liter, giving us 1.76 grams per liter. So, the outlet biomass concentration is 1.76 grams per liter. Now, let us consider the second option. Here the second option, there are two reactors which are connected in series and the reactor volumes are 50 liters each. So, the calculations which have to be performed for the first reactor will be exactly the same as what we had done in the previous example, and the only difference is the reactor volume is half. So, we calculate the mu appropriately and we will perform all these calculations which we did earlier to get the values for S and X. So, if you were to perform these calculations, you would get the values of S as 6 grams per liter, which is the S1 leaving the first reactor, and your X1 would be 0.8 grams per liter. I am not going to perform these calculations here. I expect you to perform whatever we have done till now and get these values and verify if you are able to get the same values. Now, let us write the balance equations for the second system. So, the second system, the reactor 2, we can write a cell balance. The cell balance would be Fx1 minus Fx2 plus Vr mu x equals 0. So, here we are assuming there is no cell death or accumulation because it is steady state. So, we have an inlet biomass concentration coming in terms of x1. So, we have Fx1 as the biomass entering the system, Fx2 is the biomass leaving the system, and Vr mu x is the biomass which is being generated. So, this would be mu x2. In this equation we know F is equal to 15 liters per hour and we have calculated x1 as 0.8 grams per liter and we know that Vr equals 50 liters. So, this leaves only x2 and mu as the two variables which are unknown, but what you see here is it is a non-linear equation. So, we might have to solve a non-linear equation to get the final values. So, we can similarly write the balance for the substrate. The substrate balance would be Fs1 minus Fs2 minus Vr mu x divided by Yxs equals 0. Here again we are assuming that there is no product formation and there is no accumulation. So, using this we get this equation. Here again we have already calculated S1 as 6 grams per liter. We know F and S which have been given to us. So, F and Vr which have been given to us, again this would be x2. So, we now again have two variables which are unknown. Yxs is again known to be 0.2 grams per gram. So, now with all the variables known, we are left with mu and x2. So, we can also calculate mu from the Monod's equation, which would be mu max S2 divided by Ks plus S2, where we have been given what mu max is given as 0.4 our inverse and Ks is given as 2 grams per liter. So, now that we have all the parameters, we just have nonlinear equations that need to be solved simultaneously. By solving these nonlinear equations, we will be able to get the values for the biomass concentration which will be leaving the system. If you were to perform this calculation, you will end up with the outlet substrate concentration as 1.35 grams per liter and your biomass concentration as 1.73 grams per liter. So, based on the system which we have looked at, the reactor with 100 liters volume and a single system actually gives a higher biomass concentration compared to two smaller reactors in series. So, with this I hope you have full understanding of how to perform material balance calculations for systems where cell culture is happening. Thank you.